our genes? Each of us is determined by the genes that we have inherited from our father and our mother, and generate a nice or balanced mix of paternal and maternal genes, except in cases like here where the genes of a parent can dominate, although we don't know, maybe this guy has the feet of his mother or the character of his mother. But in reality, we are a lot more than just our genes. If you take these people here, they have exactly the same genome. They are monozygotic twins, so they come from the same egg. And even though they have the same genomes, they are very different. And when one may be lean and healthy and would not develop any disease, the other may have developed metabolic disease and be obese. Another example here is that one may be a balanced person with normal behavior, while the twin brother may be aggressive, violent, may become schizophrenic or have depression. So this is because these people have been exposed to different environmental factors. And there are multiple environmental factors that can influence us, starting from what we eat, to the experience that we have throughout life, whether stressful, traumatic, or whether also positive experiences, or experiences linked to the social rank that we have. They can be drugs and chemicals or pollutants, the environment, or whether we exercise and our lifestyle, or the exposure of radiation. And all these elements influence our genes, or the way our genes are expressed or are active throughout our life. And it is really indeed throughout the whole life. From the very beginning, germ cells, whether male or female germ cells, are quite sensitive to the environment, and the embryo, postnatal, and also the young child, or throughout adolescence until adulthood and old age, all these stages have some susceptibility to environmental factors. And if you consider that these germ cells are also can also be modified by environmental factors, then this leads to a transmission across generations. Indeed, there are very prominent human examples of transmission of the effect of the environment in human. So there was a series of studies done after a Dutch famine. So it's a famine which happened in West Holland during Second World War due to a Nazi embargo, and it concerned a population of people who was well circumscribed. And it was quite easy to determine how much food and the calorie intake of these people. And the study was done essentially on pregnant women. And if you consider that a pregnant woman exposed to a lack of food will be affected with this lack of food, but also her baby, but as well the germ cells that her baby carries and who will give rise to the following progeny. And this study showed that people across 2 generations, so in the second and the third generation, had higher incidence of cardiovascular diseases, diabetes, and obesity. Another example more in the psychiatry is that exposure to traumatic experiences, violence, abuse, whether physical or sexual abuse, neglect or abandon in early life, or the loss of a parent, for instance, can lead to a number of psychiatric disorders like depression, personality and mood disorders, schizophrenia, antisocial behaviors, addiction, or even suicide across life, and that these symptoms can be also often observed in the following generations. And indeed, this led to the recognition that not only the genes, but really the environment is also important. And there were, hundreds, if not thousands, of genetic studies searching for the gene for depression or the gene for schizophrenia. And these studies have all failed to identify such genes, just because it's really the interaction between our genes and the environment, between our genetic background and epigenetic mechanism. So what is the interaction and what are epigenetic factors? So behavior, if we take behavior is controlled by the brain, and brain is made of brain cells. And you have a neuron here, which with the cell body and the nucleus, and the nucleus contains chromosomes. Only one is represented here, but we have 23 pairs of chromosomes, including the sex chromosomes, XX for women and XY for men. And the chromosome is made of the chromatin, which contains the DNA. So you see the chromatin here, which is a bit opened to see the structure, and it's constituted of the DNA, which is wrapped around protein histones. And you see the DNA is what made the genes. And you see that genes, they are in succession on the chromatin, and you may have one gene here and a second gene here. And chromosomes typically have several hundreds, up to 2,000 genes. X chromosome has 300, and the Y chromosome has 50. And the epigenetic mechanism, so maybe in analogy to the computer, the genes would be the hardware. But the hardware on its own is quite useless. You need softwares. And the softwares are the epigenetic mechanism. So we develop these softwares along our life, and these softwares are influenced by the environment. We may inherit some softwares or pieces of softwares from our parents, which may be good or bad, but which together with our own softwares will determine the activity of the genes. And these softwares are epigenetic mechanisms, and they include DNA methylation. So it's a mechanism which consists in adding a methyl residue onto the DNA. This is histone modifications. So there are also small modifications which are added on the proteins here. And there is a third one, which is newer, is non-coding RNAs. So there are RNAs which are produced from genes, but which are rather in the cytoplasm. And it's these mechanisms which together determine whether a gene is active or not. And these mechanisms can be influenced by our environment. So this is now for the behavior, for the physiology of a person or an individual itself. Now, for the transmission of characters which are acquired due to exposure to certain environments, the only link between generations are germ cells. So if an individual which carries germ cells is exposed to environmental factors or trauma in early life, and this leaves prints onto germ cells, this will alter the health and the behavior of the progeny. So germ cells, indeed, I'm showing here a male germ cell, do contain genes and epigenetic mechanism. So you may notice that the chromatin in sperm cells is a bit different because it's such a tiny cell that it needs to be compacted, and protein histones are replaced by protamines here. But there are two major epigenetic mechanisms which are known to occur in male germ cells. This is DNA methylation, so exactly the same as in other cells of the body, and non-coding RNA. So now, to demonstrate, I mean, if you hypothesize that the environment may modify this epigenetic mechanism in the brain and in germ cells and ultimately lead to psychiatric disorders, that will be transmitted across generations. You cannot really test this hypothesis in a causal way in humans, so we used an animal model, a mouse model. So we developed a mouse model of heritable traumatic stress. So it's a manipulation which consists in... So here you have a mother with her pups, so it's a regular type of raising her pups. And here we have the manipulation, which is an unpredictable maternal separation. So we separate pups from their mother anytime during the day. So they don't know this. The factor of unpredictability is very important to make the stress really traumatic. And the mother, at the same time, is stressed unpredictably on different stressors. So we do this on the first generation. And we wait until the animals are adult, and we breed them down to the third generation. And we test the behavior, and we do epigenetic analysis on each generation. And being exposed to such trauma during early life induces depression in mice. And depression is behavioral despair. So it's quite easy to test in mice. One task, which was developed in the 70s, and it's still used to test antidepressants, is called the Possold forced swim test. So you put the mouse in a glass of cold water, and the mouse will swim and try to escape. A mouse which is depressed will hardly swim and will start floating by despair. So we measure the time the animal floats and the time it takes to float. Usually they start floating quite immediately. The control mouse may ultimately float, but after fighting and trying to escape. So we observed that in the animals which were subjected to stress, so the F1, they were more depressed. They were depressed, and these depressive behaviors were transmitted across generations. And we still have our desperate guy here. So if now we try to make the correlation with epigenetic mechanism, the mouse is a fantastic model to do this because you can extract the brain, you can extract germ cells, and look at genes and look at epigenetic marks. And indeed, what we observed in the animals which were subjected to this stress, as well as in their progeny, is that there was aberrant DNA methylation on cells in the brain and on germ cells. And there was also an excess in non-coding RNA in these cells. So, which suggests that maybe being exposed to this stress altered the regulation of epigenetic mechanism, and that this alteration may be responsible for the behavior. Now, to really prove that this is the case, I mean, the best way is to mimic this alteration. So mimicking aberrant DNA methylation in this case is not possible because it's complicated. There is too much DNA methylation on some parts of the genes and not enough on other parts, and it's just not possible to recreate exactly these changes. Now, recreating, mimicking an excess in non-coding RNA is something more tractable, and that's what we did. So what we did was to take a fertilized control egg. So it's an egg which has already been fertilized by a sperm cell. And we take the sperm cell from stressed males, but we extract just the RNA from these sperm cells. We don't take the sperm cells in an entire sperm cell, we just take the RNA. And the RNA, then we inject into this fertilized control egg. So if it is responsible for the phenotype, for the altered expression, altered behavior, we should see it in the resulting progeny. And the way we do it, it's under a microscope. So we take the egg that you see here, fertilized egg, and we have a needle, injection needle with the RNA. And then once the eggs are injected, we take them, we do this by surgery, we re-implant them into pseudo-pregnant females. We wait 3 weeks until the animals are born, and then when they are born, we wait until they are adults, and we test the behavior. And what we observed was that these animals resulting from the eggs, which were injected with just the RNA from sperm cells from traumatized males, they were depressed as the males that we used to extract the RNA. But not only them, but also their progeny. So if they are bred with regular, normal, wild-type animals, these animals are also depressed. So we should suggest that these epigenetic marks, or RNA, does carry a memory of the trauma and is a good mediator, is a mediator of the transmission across generations. So I'll finish with a few perspectives for humans. So this type of study, which, so we are interested in stress, but there are other labs in the world who are looking at the effect of diet, the effect of plastic or exposure to chemicals. So identifying the biological or the epigenetic markers which are linked to this type of diseases is very important for human beings because they provide a new understanding of diseases which are induced by environmental factors. There are many such diseases. For instance, cancer, most cancers are from environmental origin. And understanding these markers, in particular RNA, because RNA are also present in blood. And in human blood, it's quite easy to measure. I mean, there are small pieces of RNA which are present in blood in mice and human. And in human, it's quite easy to measure the level of microRNAs in blood. And it's also important type of findings because it provides a novel view on evolution. It's fast evolution, but it's a way for people to adapt to the environment, whether positive or negative. We also show that animals exposed to traumatic stress can develop some strategies to be more efficient in certain conditions. So this can have some advantage. And this is a way to evolve quite rapidly without changing the hardware, without changing the code of the genes.